What are the most promising technologies that could really shake things up for health span and lifespan in the next 5 to 10 years? Here are my thoughts. What are my criteria? What makes an intervention a contender? First, impact. It has to genuinely move the needle on health span and lifespan. Aging is complex, so I'm skeptical of any single magic bullet molecule. Second, a path to the clinic. A straightforward route to human trials is key. We need to see a realistic path that will bring the intervention to market. And three, active development. There needs to be a dedicated organization pushing it forward. I'm a believer in eventual radical life extension, but I think the next wave of interventions will focus on extending health span and lifespan, while those more advanced approaches are still being researched. Lifestyle is always the foundation. It's crucial for a good health span, but I am not convinced it can drastically extend maximum lifespan. I see these interventions as add-ons to a healthy lifestyle. So here are my top picks, in no particular order. I'll give you a quick overview now, and then we'll dive into clips from the interviews where experts explain the science and the path to the clinic. Thymus regeneration. Dr. Greg Fahy at Intervening Immune is working on this. Our immune system weakens as we age, so rejuvenating it is a recurring theme on my list. A strong immune system is vital for health span. It helps clear out cancerous and senescent cells, fights off infection, and keeps chronic inflammation in check. Dr. Fahy is using approved ingredients with known safety profiles, has already seen success in human trials, and is moving to the next phase. Exosomes and factors from young stem cells. I'm grouping these together. We're talking about the secretome of placental stem cells and factors from young pig's blood, from Dr. Hans Keirstead and Harold Katcher, respectively. Extracellular vesicles from young cells seem to have a systemic rejuvenation effect. Doctors Keirstead and Katcher have both found innovative ways to scale up production, which is a big hurdle with these type of therapies. Mitochondrial transplantation. Mitochondria are the powerhouses of our cells. They're constantly renewed, but their DNA degrades over time. Replacing old mitochondria with fresh new ones could give our cells serious energy. Mitrix seems to have a good handle on the safety aspects and is planning human trials within the year. Immune system rejuvenation. Dr. Alessio Lanna's work is super promising and involves a simple peptide which regulatory bodies in the UK consider to be a small molecule. Human trials are also planned later this year. Can we talk a little bit about why the thymus is so important, and why does addressing the thymus have such a positive effect on health span and lifespan? Because I think I didn't fully appreciate that kind of last time we spoke.
Yeah, the thymus is essential for longevity because it is the center of production for your T cells. The T cells are the adaptive part of your immune system. So you have more evolutionarily ancient aspects of immune system like natural killer cells and so forth that are essential, but not as sophisticated as your adaptive immune system. And it's really the adaptive immune system that suffers the most with aging. So you retain B cell activity reasonably well as you get older. You retain natural killer cell reasonably well as you get older, but your T cell activity declines catastrophically as you get older, particularly as you get to be older than 60 or so. And so I've done some indirect research on the significance of this by looking at statistics compiled by the Center for Disease Control in the United States. And if you add up the likelihood of death from immune system issues such as cancer or infectious disease, your risk of death as a proportion of the total risk of death goes up dramatically with age. So say, for example, at the age of 25, your risk of death is only affected by about 3% in terms of immune system issues. But as you get to be 50, it's more like 50% or 30%. So the curve is exponential. So as you get older and older, your risk of death from immune failure issues increases exponentially and probably turns out to account for at least half the deaths, if not more than half, as you get over the age of 60. As a matter of fact, the ability of your immune cells to recognize foreign antigens declines by about 98% before the age of 80. And so you have to have immune system function in order to stay alive for the long term. And this is a long-term process. The thymus, located here in your breast, under your breastbone, is the center of production of T cells. And that center of production begins to shut down when you go through puberty. The T cells that are produced last for a lifetime, but not for a complete lifetime. They last until about the age of 62 or so, and then they begin to disappear. And that's about the age at which we all begin to die. So that's probably not a coincidence there. So I think that if we do everything else we want to do about aging, we may have great success in many respects, but we'll still die more or less on schedule, because without an immune system function, even young people die. Early on in the AIDS epidemic, immunodeficiency virus killed a lot of very young gay men. And it's because you just have to have an immune system. So I think that we need to have more focus on the thymus than has traditionally been the case. And I think if we do, we'll not only live longer, but we'll live more healthy as well.
Right. So the thymus actually matures the T cells, right? They get produced somewhere else. You still need the T cells, but
you have progenitor cells that are manufactured in the bone marrow, and they're released into the blood, and they develop into different lineages. Some of them go on to form B cells. Some of them go on to form T cells by means of thymus education. So they develop from these precursor cells in the thymocytes in the thymus, and then when they're fully mature, they're released back into the bloodstream as real T cells.
What is your plan to turn this into a therapy that can be delivered?
Yes. So we actually have multiple plans that will be implemented in a kind of a layered fashion over time. So every company has kind of a pipeline of products along the way. We have pipelines as well in mind. Some of them are just laboratory ideas right now that will be developed in the products. Some of them involve known technology that has been abandoned by others that we can pick up and use to our advantage. Things that have already gotten into human clinical trials successfully, but never panned out for other purposes that can be used for our purposes, or orphaned drugs kind of thing. But just with the straight protocol that we're doing now and minor, improvements on that protocol, we already are going to be experimentally launching clinical activities in the UK before the end of the year.
Wow.
Yes, there's a group of very bright doctors who are so enthusiastic about what we've done that they just want to try it. And so we're going to try this on a trial basis. So we have developed software. So as you mentioned earlier, there are three main components of the treatment. There's also some nutrients that are included that we've talked about in the past. And generally speaking, it turns out to be much, much, much more complicated than you would imagine. Figuring out how to adjust the dose of each of these three major components as you go along, because I've actually we've put together an expert system that takes into account over 100 different factors that go into just choosing the direction of each one of the changes in these three things. You wouldn't think it could be that complicated, but actually it really is, because so
Mm.
many things are going on. We have to pay attention to a lot of different things that are going on. We measure all kinds of uh, blood levels of different items in the blood, and we try to look at the person holistically and what's going on with them, and try to figure out what's the best course for them. And there are contradictions. There are indices that say increase this, and there are some things that say decrease that, and you have to decide what's more important and what's the hierarchy and all of this. And to ask a naive physician, even a good, smart physician, to figure all of this out on the fly, it's unreasonable. They're not going to do it. It's not going to happen correctly. I spend tremendous amounts of time on this myself, and I've been doing this for years. So we've been developing the expert systems to make this possible, and we're going to be testing this more and more. So far, the expert system has made appropriate calls every time that we've tested it recently, and that's going to be necessary for deploying this. It's also going to be helpful for us to retain control over the treatment, because doctors really need to work with us in order to do this correctly. And we're not going to license the use of this technique to clinics that aren't doing it correctly. So if the clinics are following our protocol with our software and reporting their data back to us so we can continuously refine that software, and we know that they're going to be maximizing efficacy and minimizing any harmful side effects. And at the same time, our expert system, which will be converted into an AI over a fairly short amount of time, will get stronger and stronger, and we'll be able to do a better and better job of expanding this. So this is the way that we can develop the treatment into something that can be uniformly applied by many different doctors, many different clinics around the world, in a way that is consistent, in a way that is correct, in a way that continues to feed back on us to make it even better. So, there are many different people. One of the things that makes it so complicated is that everybody's an individual. People respond differently to the same stimulus. And sometimes it's not apparent what's going on for a while. And sometimes the body changes, the body actually adapts to some of the treatments, and you have to adjust for that as you go along. So it gets very, very complicated. So you need to have a uniform way of dealing with all of this so that you get a uniform result. And you need that sort of thing to minimize variations in outcome that blur the statistics, that make it harder to get FDA approval. So this clinic that we're working with now is operating in a jurisdiction in which they don't need regulatory approval. In the United States, you need the FDA to approve this. The FDA has looked at our treatment and decided that even though all three components of our treatment are vanilla as far as the FDA is concerned, and the FDA is not actually worried about our treatment. They're not, it doesn't bother them, but they consider it a new drug entity. And I have to say, I agree with them about that, because the combination does things that none of the three components do by themselves. So it really is an interaction between these three things that we're taking into the clinical market in the United States. So I have to agree with the FDA that it needs to be seen as such. And so we're going to be going down a regulatory pathway that the FDA will approve. And meanwhile, we'll have some jurisdiction in which we can operate more freely and expand our range of experience. And we can bring all of this together so that whatever we submit to the FDA in the end will be informed by the largest number of experiences possible. But meanwhile, it won't be held up indefinitely, by the regulatory process, because it can be sort of introduced gradually and slowly in different ways as we feel our way along. So I think this is an appropriate way to go. It kind of strikes a balance between the fact that people have an immediate need for aging interventions and the fact that you don't want to go headlong into something that might have complications that you haven't discovered yet. So, I think this sort of gradual, thoughtful, scientific approach is appropriate and will get us into the clinic. And if we can make this a scalable process in spite of all the complexities, and we think we can do that, then it can be expanded to millions and millions of people. And if we're doing this in a way that brings revenues back to us as a company, we will benefit, and even as the patients, benefit all over the world. So that's kind of the plan.
Happy New Year. How are your New Year's resolutions coming along? If you're like us at Modern Healthspan, you're probably prioritizing your well-being this year. And we know that good sleep is absolutely essential for feeling our best. And if I had to pick just one thing to boost my sleep and overall health, it would be magnesium. And not just any magnesium. Specifically, I'm talking about Magnesium Breakthrough by BiOptimizers. Magnesium Breakthrough is formulated with 7 different forms of magnesium, each with its own unique benefits. From improving sleep quality and boosting brain function to supporting a healthy metabolism and managing stress, magnesium plays a role in over 600 biochemical reactions in the body. Even if your New Year's resolutions aren't specifically health focused, it's tough to achieve any goal without good sleep and effective stress management. So why not give yourself a helping hand? Go to bioptimizers.com/modern and use the promo code MODERN10 to receive 10% off your order. Plus, when you subscribe, you'll unlock exclusive discounts, free gifts, and ensure you have a convenient monthly supply. Wishing you a well-rested and successful 2025. So, Dr. Katcher, so your team recently published a paper on bioRxiv, so reversal of biological age in multiple rat organs by young porcine plasma fraction.
Right.
Some updates to your work on E5. So could you talk a little bit about, so what is new in this latest paper? What's kind of the updates in it?
Well, as you detailed in your own podcast, I revealed the secrets of E5, which is basically 2 things. One is that it's primarily nanoparticles like exosomes that are the basis of rejuvenation. And that completely disagrees with what was previously thought by people like the Gun Boys and Saul Valeda, etc., Amy Wagers, who all attributed anti-aging of blood plasma to small molecules, proteins. I never believed that was true for some obvious reasons. For example, any single gene can be mutated, and the lack of that gene or the over secretion of it should produce vast differences in lifespan, and that doesn't happen, except for lamin A. And even that's a question of alternative splicing or alternate splicing. And the second thing is, and I consider this really the most important thing because it makes it a viable option for all of humanity, is that it was porcine plasma, not human plasma. And that leads to 2 things. One is the fact that porcine plasma is apparently, or rather the nanoparticles, the exosomes, etc., in porcine plasma don't have any immune reaction in rats or for that matter in human beings. In the one instance that I successfully tried it, which is my own hand.
We want to make the reproduction of Harold Katcher's experiment in a authorized facility, a certified facility to make preclinical trials in an international authorization level. What means that it's a place that the big pharma or any other international company do their trials in rats, and those results will be certified not only by Brazilian regulation authorities, but international and other countries' regulation authorities like the FDA or the EMA in Europe or China's institutions. I mean, here in Brazil, in the state of Santa Catarina, there is an institution that exists since 2014, is relatively new, and is equipped and authorized with the highest standards in international authorization, and they make services for the international pharma companies. So if we do that, the study of the rats there, they will make all the safety studies in the rats. These studies are necessary to allow human trials. But as we will publish everything, everybody in the world could use our data to try this therapy in humans, at least in compassionate patients. In Brazil, and I believe that in other countries it's the same, the compassionate patients are human patients that are dying, that they have a disease, and there is no cure for that disease, and that there's not treatment, and they are dying.
We would like to start the experiment this year. So if we have the funding by then. If not, next year, the latest, we think it's possible to do it like in the second semester, right, of 2025.
Yeah, if you had the funding right now, for example, I think 4 or 5 months, we'll be able to start the experiment. But we think that this experiment with rats is the hardest. I mean, that's because in our timeline, we give 2025 and 2026 for the rats experiment, and 2027 for the non-human primate experiment, and 2028 for the human.
So when we look at modulators of aging, you said that the underlying mechanism is immunosenescence in many cases. Could you get into that in a bit more detail?
Sure. Yeah. The immune system in many ways is the primary controller of your aging. It's it's just a fact that most of the manifestations of aging are actually immune mediated. Muscle atrophy, metabolism, arterial stiffness, neuroinflammation, wound healing, the loss of the adaptive immune response. These things are all immune mediated manifestations of aging. And the importance of the immune system in your health span and your lifespan can't be overstated.
So how does the immune system change with age? What kind of changes do we see?
Basically your immune system gets old and angry with age. It's 100% of humans develop pro-inflammation, just a chronic state of inflammation within their blood systems. And that is largely mediated by the fact that you have fewer progenitor cells. The young cells of your immune system are born very frequently when you're young, and they're born very infrequently when you're old. And as it is the young cells, the progenitor cells that secrete, you have fewer secretions in your blood, because when you're over 27 years old and you're birthing your immune system less frequently, you have a dearth of these, what are referred to commonly as nursing factors, hundreds of growth factors and cytokines and chemokines, exosomes, a few other things that are released by young progenitor cells. Your mature cells do not secrete. It's your young cells that do. And because you birth your immune system less frequently with age, you have fewer progenitor cells of your immune system, and therefore, with age, you have fewer of a secretome, these nursing factors. And what those things do is DNA repair. They are responsible for rolling of the immune system out of your blood and into tissues. And that's a big deal. 80% of your immune system resides outside of your blood. So these extracellular matrix and cell-cell adhesion molecules and cytokines and chemokines and a few other things that cause the rolling and the attraction and movement of these immune system cells, you just have a lot less of them with age. So at Immunis, we developed a method of taking human stem cells and making them, for the first time in the world, to a young, defined progenitor cell population, human, of course, that makes 440 active factors that modulate the immune system. Just like your immune system precursors made tons of it when you were young, we can basically make that in huge bioreactors now by the billions. Basically take young human stem cells and then make them into these young progenitor cells and define their differentiation perfectly so that we have a secretome that is identical batch to batch. And that secretome is every factor relevant to human immune system development and health, the very natural factors that you're full of when you're 27 and you have quite decidedly less of those things as you age. We found a way to make them. And in both animal models and now, super exciting, in humans, we see that it's both safe and efficacious. And then we applied to the United States FDA and we were awarded a phase 1/2A, 9 outcome measures of efficacy and about a dozen outcome measures of safety. This is a first in human test, and it's been going on over the last year at the University of California Irvine Medical Center, UCI Health. And I'm just absolutely delighted to say that in older humans that are immobilized, so they can't exercise. This is immobilized elderly humans. We're seeing their muscles grow. We're seeing them get stronger. The muscles get stronger and the whole limb getting stronger. We're seeing in a functional quality of life measurement recognized by the FDA and CDC as a quality of life measurement. It's called the 6-minute walk test. That they are statistically significantly improved, 20% faster and further walking within 6 minutes. Their pain is dropped 50 to 70%. Their immune systems, the chronic state of inflammation of their immune systems is dropped by about half. That's extraordinary. 3 months after treatment, these 60 and 70-year-old people have an immune system that is characteristic of someone decades younger than them. Their basal state of inflammation is dropped dramatically. So it seems to be working and no side effects, which is wonderful. So this is not published, the human work yet, because that just rolled out over the last month. So we're collecting all of the rest of the phase 1/2A data through to August of this year, when it'll completely end. And we have prepared a phase 2 already because the data are without any side effects, no adverse events, and we're having tremendous efficacy. We have already gone ahead and written the entirety of the phase 2 clinical trial in which we're going to test both muscle and metabolism now in a much broader range of humans that have a broad spectrum of metabolic and muscle disorders. This is called a basket adaptive clinical trial that has a lot of people in the beginning, and it's expensive because there's a lot of people, but it's de-risked because there's a lot of people, and you can adapt it to the responders in order to ensure that you're really focusing on what works best and giving humans that, but you're also taking a broad look at many outcome measures in order to try to capture as many benefits as we can. And as I've relayed, the benefits of a healthy immune system are diverse. So that means we need to capture a very broad set of inclusion and exclusion criteria. It's pretty amazing. It's a lot of fun.
Of course, there's been all these theories of mitochondrial aging for 40 years, and people have had all kinds of things where they say, oh, it's Ross or it's various other things that are causing mitochondria to age. But the thing to remember is that the mitochondria itself is rebuilt every 100 days. And so the mitochondrial membrane, it's rebuilt anyway. So it's hard to imagine that that really is the cause of aging. The thing that is eternal, that is not rebuilt, is the mitochondrial DNA. The mitochondrial DNA is a tiny little ring. It's similar to a bacterial DNA. Generally, there's about 5 of them per mitochondria. So they're actually spaced along the inside of the mitochondrial membrane. And they're constantly, and they're tiny. They're 16.5 kilobits of data, kilobases, whereas our nuclear DNA is 8 billion kilobases of data. So obviously the mitochondrial DNA is this teensy-weensy little thing. And all it does is it just runs, as far as we can tell, it just produces a couple key proteins that are really need to be produced inside. It would be too inefficient. Remember that mitochondria are designed for optimal efficiency. If you're a rabbit and you're running to try to get away from a wolf, you live or you die based on the efficiency of your mitochondria. So they are optimized every way possible. And so one of our philosophies is, however mitochondria are built right now is probably at 99.99% as good as they could be built. They've been so well optimized by nature. So,
so you've looked.
So you were asking about what goes wrong.
Yes.
So those little mitochondrial DNA in there, they're replicating constantly, of course, because as mitochondria are replicated, the mitochondrial DNA are replicated. And the problem is that as they're replicated, they begin to get transcription errors. That is our theory, and quite a few other people now. The DNA itself, the mitochondrial DNA, acquires like bad sectors on a hard disk. Okay. If you remember the old hard disks where you could look and you could see all the little black dots where the bad, and the computer has to work around all that, right? Well, that's our theory, is that there's bad sectors on the mitochondrial DNA because it is such an intense environment. It's a factory. It's very hard to do quality control. It's very intense. This is happening. It's like it's hot. It's you could imagine trying to do something careful when you're inside of a steel mill and there's fire everywhere. I mean, it's a very difficult environment. And so we start out, when we're babies, we start out with a huge complement of absolutely perfect mitochondrial DNA. And then as the decades go by, they get more and more and more degraded from these replication errors. And according to our research, by the time you're about 95 years old, they're shot. They can't support you anymore.
Okay. So speaking of humans, you have started this trial. And I think you call it like the 130-year project, because you're aiming to beat the 122. That's the current.
We are aiming to beat the 122-year lifespan. Exactly. And now, obviously, we won't know that for a while. The oldest person we have is 90 years old, who's a volunteer. So it may take us 40 years to prove it. But if we can show, I don't know, are you familiar with the Healthspan X Prize?
A little bit. Yeah. If you could provide a little bit of background on it.
Yeah, the Healthspan X Prize was sponsored by the Saudi Arabian government, and it's the X Prize Foundation is running it, and the goal is it's a $101 million prize, and the goal is to show that you can take 10 people and make them 20 years younger. Okay, and of course everybody says, well, what exactly does that mean? It's not like you can just put somebody on a treadmill and get some exercise and say, oh, they're 20 years younger. That doesn't count. You have to figure out a way to prove that you've made them physically and genetically younger, right? Which is challenging. But that's our goal. Our goal is to show that we can take our 20 volunteers and make them 10, 20, whatever years younger, so that they have increased muscle mass, their brain improves, their cognition improves, their heart regenerates, Their immune system gets better, that they are literally younger. Okay.
Okay. And when are you going to start this trial?
We've already started the early phases. As I said, we've gotten the volunteers. We decided to get a group of volunteers early because we wanted their help in kind of helping us determine how to do this. And you know, when you're working with 80-year-olds, you're getting people with a lot of experience, right? So I have a couple really, really good doctors who are in their 80s, who are still practicing doctors, who are experts in gerontology, and I'm getting the benefit of their experience for this project. So that's really exciting. The goal is to start it in a year to a year and a half from now. That would be 2025. But we have to, we are in the process of raising the money to do that. It's, you need significant funding to do something like this. This is not a biohacker project. You need a lot of big bioreactors, and they're very expensive.
Right. And it is the funding, it's not approval from any regulatory agencies that's the limiting factor.
No, we're probably going to do this in either Japan or the Bahamas. Because both of them have specific mechanisms in their regulatory rules that allow for these sorts of trials. Japan, for example, if you get through the phase 1 trials, which are very rigorous, I mean, you have to do a good job. And we're going to do, we have some of the best scientists on the West Coast and East. We have some of the best scientists from all over the world who are going to be supporting this project. So we have to do this very high quality. We want this data to be usable by anybody. This is a, we want this data to apply to the whole human race. Okay. So we're doing it very well. In Japan, if you go through the phase 1 trials and prove safety, then they'll let you actually sell it to the general public as part of the phase 2 trials. They have a different system there where they use the data from early customers as part of the trials. And that's also a way that makes it easier for companies to get past the funding hurdles, because they can sell things early.
Santicel, we focus on this, I would say, unanswered medical question, which is how we modulate the immune senescence of T cells. If you think about other pathways, such as regulatory T cells or exhaustion, immune senescence has been largely overlooked, meaning that to date, there is not a drug that can actually target these immune senescent cells to rejuvenate them. So immune senescence can be defined as all sorts of bad things that happen to immune cells when they grow older. And this causes a bunch of problems, starting from a lack of responses to vaccination, the inability to control infections, the surge of cancers with age, and also nowadays this has been connected with neurodegenerative disorders, for instance. However, we're moving forward to the awareness that if you regulate the youth of a single cell type, which is the CD4 T cell, you can control the overall lifespan and health span of the organism. And this is what our data show. In order to do this, we've been focusing on a complex of proteins that are characterized during my university years before founding Santicel, when I was a PhD student at University College London, which is called the SMAC. The SMAC is a complex of stress proteins bound together that dictate aging phenotypes to the T cells. So, by disrupting the SMAC with the first-in-class compound, we can impose a rejuvenation on these cells, and we can change their aging fate. We can reprogram them to give them new functions.
The human trial is starting next year.
Next year, yes.
And do you have any idea on how long is it going to take? And
the phase 1, which we expect to be very smooth, because another amazing thing about these compounds is that in terms of the NOEL, the NOEL overlaps with the maximum feasible dose, meaning that we observe no adverse effects up to the maximum feasible dose we could administer to the animals, which is over 200 times or 300 times the clinically approved dose. So we have the preclinical dose at which we see the effect of interest. We're using the compound at a concentration of 0.0 0.05 mg kilo. Think about other therapeutic compounds that have been used at 20 mg kilo, 30 mg kilo. So very short amount of the compound, very large therapeutic window. So the phase 1 for safety shall be very smooth. So we expect it to be ended in less than 1 year and then get into phase 2.
Interesting. And so in terms of approval, what you're looking to do is to show that you get better response from vaccines.
Well, it's a complex topic, I would say, because so we will start the testing with vaccination, to perfect vaccination. We will also have a group of people that do not get vaccines but only get the rejuvenating compound to see whether they can be protected in the future from unrelated challenges or from infections and so on. Obviously, we need something to measure. So in order to measure something, the easiest thing is to measure the response to the vaccine. So of course, we will be relying on the vaccine response, but we will also, on a side, we will monitor the ability of people who are on the rejuvenating compound, which is called DOS, who are on DOS, to cope with unrelated challenges with or without vaccination. So we do want to have one arm of the trial without the vaccine, just with the DOS, just to see whether those people, for instance, how many infections we report during the winter, not only for infections, any sort of infections, and they can be, of course, they can be not only reported, but can be documented in the laboratory. So we want to see, because we think that by rejuvenating these T cells with the DOS compound, these people will be better off, for instance, against seasonal flu and so on, even cancers. But of course, we have a short amount of time. So we have only the observation. Of course, the observation will be short, like 1 year or 8 months or 9 months, but it's still clinically relevant. But obviously, the decisions will be initially based on the response to the flu vaccination. But we are very much aware that this is just the beginning and so much more can be done.
There are other cool technologies out there. For example, gold nanoparticles from Clean, and the mind-blowing work Mary Lou Jepson is doing at Open Water. One notable absence from my list is partial reprogramming, like what Dr. Sebastiano is doing at Term Bio, also Altos Lab, and others are working on. The current success rate with Yamanaka factors is still really low, under 1%. Retrobiotech is using AI to improve this, which is awesome, but the regulatory pathway looks tough. It just doesn't seem to me like it will be ready anytime soon. So what do you think? Do you agree with my list? What technologies are you most excited about? Let me know in the comments. Thanks for watching, and I wish you all well.